So we're going to do now is our final lab of the semester. And it's basically going to be looking at some of these different breakdown processes along the different levels of the digestive path and see how they're affected by different kinds of conditions. So, in this lab, we are going to be kind of mimicking the processes that happen inside your mouth or in your stomach or in your intestines, but we're going to be doing it what's called in vitro. What does it mean to be doing experiments in vitro?
Like within?
Say it again.
It's within something?
It is within something, but this is as opposed to versus in vivo. These are two kind of different approaches to doing experiments, particularly when you're trying to understand what's going on in a living thing.
In a Petri capsule? That's what I thought.
Yeah, so vitreous means glass. So in vivo is if you're actually studying the processes within the living animal. In vitro means in glass, it means like in a test tube or something. It's like, let's look at the processes, but not in the body. Let's kind of try to simulate things in a, because doing things in the body is tricky. It's very difficult to control, very difficult to measure. So it's very common to kind of try to simulate things in a test tube, for instance, and use that to get information about the processes. So that's kind of what we're doing in today's lab, is we're going to be looking at these processes of enzymatic breakdown, but we're not going to actually do it inside a person. We're going to actually do it in a test tube. And we're going to be looking both at the breakdown of carbohydrates, breakdown of proteins, the breakdown of lipids. In each case, we're going to have enzymes that can do the breakdown. For carbs, we have salivary amylase. That's the amylase in your saliva. Normally, we'd have someone in your group spitting into a little tube, but in this case, Becky did. Protein, we're going to have pepsin, which is the stuff that is made in your stomach. For lipids, we're going to have, it's called pancreatin, but it's basically the lipase made by your pancreas. So we're going to have enzymes which just come in. This we're going to get from somebody's spit. These we're just going to get from the chemical supply house. We obviously need our example of a carbohydrate. For carbohydrate, we're just going to use starch solution. For the protein, we're going to use egg white, boiled egg white. For the lipid, we're going to make life easier, we're going to use cream. Cream is already emulsified to some degree, right? Cream isn't just oil. It's kind of like all the milk fats kind of suspended in kind of a watery stuff already. Like that's like if you're making butter, you end up with like the whey and all that. So it's already emulsified to some degree, but we'll see, we can do more. So this is kind of how we start. And we're going to be able to change conditions. We can change pH where the enzyme is working on the substrate, right? These are the substrates. This is our enzyme. We can change pH by adding or subtract, adding hydrochloric acid or adding sodium hydroxide. We can change when we're doing lipids, we can add or subtract bile to the mix to see how that affects things. So we're going to look at each of these three breakdown processes and then alter different conditions and see how that affects the breakdown. Obviously, in all three cases, we're going to need some way to assess breakdown. So I have my question of, how does pH or how does this or that affect the breakdown of carbohydrates by amylase or proteins by pepsin or lipids by pancreatin? I need some way to assess how much breakdown actually happened. For the egg white, it's going to be easy. Egg white is just going to be a visual, did egg white dissolve? Right, I'm gonna take some egg white boiled, like it's hard boiled, and if it's digesting, breaking down, it's gonna just go into solution. It's gonna start disappearing. So if the egg white is dissolving, that's evidence that it's breaking down into these smaller molecules that are going into solution. For the cream, for the lipids, I am going to add a pH indicator to the cream. So we're going to have what is blah, la, 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 la. God, my brain is, I'm spazzing on the word. Litmus cream. So litmus, if you haven't used it, litmus is something that turns red if it's low pH. And it turns blue if it's alkaline. So when you first add the litmus powder, this is just this powder, when you add it to the cream, since the cream is not alkaline, since the cream is not acidic, it's going to be bluish. If the fats break down, what do fats break down into?
No, sorry, that's carbohydrate.
Glycerol and fatty acids.
Exactly, glycerol and fatty acids. So the more that the fats are breaking down, the more we're going to have fatty acids, which means like, oh my God, the pH is going to go down, which case it's going to start turning more red or, probably more of a purple or something because, blue turning to red, kind of going through purple and pink. So the more breakdown I see of the fats, the more kind of pink. It's more actually pink than red. The more pinkish it gets. So that's going to be my way to assess the breakdown of fats. If it stays blue, the fats aren't breaking down. If it's turning pink, that means the fats are breaking down into fatty acids. Finally, for carbohydrates, we actually have a test for both the substrate and the breakdown product. We have Lugol's iodine. This is kind of purple-black in presence of starch. So I can add Lugol's iodine, and if it's turning purple-black, then the starch is still there. It did not break down. Right? The more that the starch is broken down, the less of a reaction I'm going to get with the iodine. I also have this other stuff called Benedict's solution. This turns color if you have reducing sugars, things like maltose. As you get to the simpler sugars, Benedict's solution will start, it starts blue. Blue if there's no sugar. And if there is sugar, you know, in this case, we can think of it being maltose. You know, it's going to go a little bit green to, you know, all the way up to kind of orange and red. So if you have any color that's not blue, if it's turning green or orange, that means we have maltose. And again, if you think about starch is just all these glucoses. You know, what do I call this? If I broke this apart and now I just have a chain of 2 glucoses together, what do I call this?
A disaccharide.
The disaccharide. And what particular disaccharide is this?
Maltose.
This is maltose. So if I start seeing the Benedict solution positive, saying the presence of maltose, that's telling me that the amylase is getting busy breaking apart the starch into smaller bits. So that's going to be my assay for the carbohydrates. I'll be able to both check to see is there still starch or is the starch gone? And I'll also be able to look for the products. Are there any products of digestion, this maltose, or is there no maltose? Right, so if there was no digestion at all, what would I expect? Again, there's the iodine versus Benedict's. Let's say there was no digestion at all that happened with the starch. What would I expect to see with these 2 tests?
It's going to be blue, dark purple.
Yeah, it would be dark purple for Lugal's iodine because that means there's lots of starch. And for Benedict's,
Blue.
it would be blue. It's a negative, meaning there's no sugars. What about if I had like total digestion? Let's say the digestion totally broke down all of this starch. What would I see?
In Benedict's solution would be greenish, orange, or red.
Exactly. And the Lugol's would stay, Lugol's if it's normally just kind of like a yellow brown. If it stays kind of that yellow brown, that means that there's no starch present. Kind of amber, maybe amber is like yellow brown. So that is kind of the basic background you need for this lab. We're going to be looking at 3 things. Like I said, we're going to be looking at breakdown of carbohydrates in the presence of amylase, breakdown of protein, in this case egg white, in the presence of pepsin, and the breakdown of lipids in the presence of pancreatin. And we're going to have a variety of different conditions. So what I'm going to do now is I'm going to share the actual lab protocol. So this is just your lab manual. So what you're going to be doing in a few moments with your breakout rooms is for each tube, for each of these 3 sections, you are going to have to come up with a hypothesis about what you're going to find. So test tube number 1 here, this is for the carbohydrate. It's starch and water sitting. Why is it sitting at a 37-degree water bath?
That's body temperature.
Body temperature.
Exactly. It's mimicking body temperature. So you're going to have to say after this starch and water have been sitting together at body temperature for a half an hour. What are you going to expect to see in those tests for starch and maltose? Test tube 2, it has starch and saliva incubating at body temperature. What are we going to see there and why? Number 3, it has starch and saliva, but it also has this concentrated hydrochloric acid that's added in. So then you have to think, what are you going to find here and why? Test tubes 4 and 5, you should still say what you expect here, but these are basically controls. These are, we'll talk about this a little more later, but basically whenever you do an experiment where you have unknowns and you have some test, you always want to validate your test, double check your test. Use something where you know what the answer is supposed to be. If I know I am testing pure starch, it better be positive for starch with Lugol's and negative for maltose with Benedict's. If I'm doing pure maltose, it better be negative for starch and positive for maltose. So you should, so I will go over that a little more, but you should at least write down what the expectation, those are controls, but you know what they're supposed to be, and you need to make sure that they are what they are, or else that means there's some problem in your experiment that's going to affect your ability to interpret the unknown results. Gastric digestion of protein. Again, we're going to have little egg whites that are all the same size when we start. We have egg white and pepsin, just kind of in water. We have egg whites with hydrochloric acid, which is going to make it, low pH. We have egg whites with an alkaline solution. Well, no, this is just, I'm sorry, this is just acid. This is pepsin and acid. This is pepsin and a base. And then this is egg whites just sitting in water on its own. So for each of these 5 things, after they've been sitting for an hour, what do you expect? Do you expect the egg white to be broken down or not? Broken down a little, broken down a lot? And again, why? Again, there's 5 little mini hypotheses. And then finally, digestion of fat with pancreatic lipase and bile salts. We have the cream. Again, litmus cream just means cream with that pH indicator in it and some pancreatin. We have litmus cream without any pancreatin, just with some bile salts. We have litmus cream with pancreatin and bile salts together. And then we just have litmus cream sitting with water. In each of these 4 cases, you want to tell me, what do you expect to see? Do you expect to see breakdown or not, a lot or a little? So what I am going to do now is first make sure there's no questions. Are there any questions about what you are about to do?
What do you mean by a pinch of bile salts?
It's just this stuff. It's like this kind of stinky brownish powder that you add.
It's an emulsifier?
Bile salts are they're the emulsifier. So if questions come up while you're doing this, just let me know and I'll come into your group. We got 1, 2, 3, 4, 5, 6, 7, 8. Oh, wait a second. 1, 2, 3, 4, 5, 6, 7. The people are like leaving, which is not good. That's actually not good at all. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. All right, so we're going to be in groups of 3 or 4. And I'm going to give you a half an hour to just go in your lab notebook. In your lab notebook, you are going to just write down for each of these 3 parts, for each test tube, for each of the 3 parts, what do you expect to find and why? You know, and again, you can work together and make sure you reach a consensus so it all makes sense. And again, but hypothesis is what and why. Like, what are you going to see and why does that make sense? And then in a half hour from now, which is going to be noon, we are going to come back together and look at what actually happens when you do the experiment. So you'll be able to kind of see the results. So again, all right. So was everybody able to kind of come up with some hypotheses that you all agreed on and kind of made sense to you?
Mm-hmm.
All right, so what we're going to do now is look at what actually happened, as well as there's some parts of the lab that, you know, are kind of asides. So we'll also have some little videos that show you some of those. So in fact, the first part of the carbohydrate lab was actually looking at just the composition of the saliva. So let me go share my screen now.
Our group didn't come to a consensus on some of the protein ones with the egg whites.
So let's discuss that briefly then. So like which ones, for instance?
It was numbers 2, 3, and 4. Let me pull it up. So number 2, egg white, 5 milliliters of hydrochloric acid and 5 milliliters of water. We're not sure because egg white's a protein, so the hydrochloric acid might denature the egg white, but that wouldn't be.
It's not necessarily going to be breaking the peptide bonds and all, right? So that's not necessarily, we don't have an enzyme there, so it might tweak things, but it's not necessarily conditions where you're going to be dissolving it.
Okay, and yeah, so that makes sense. Now for number 3, then there's hydrochloric acid, but there's also a an enzyme, but the hydrochloric acid is going to mess with the pepsin, and there's not going to be a reaction. Or
no, think about what environment does pepsin live in.
Oh, yeah, okay, it's very acidic. Okay.
Right, pepsin is actually more likely to be denatured at neutral or alkaline because it's, you know, the way it's evolved is to have its perfect shape when it's really acidic. So
okay, and then so then number 4, what is it? Number 4 then would be that's a base, sodium hydroxide. Then that's going to mess with the pepsin, and then there's not going to be a breakdown.
exactly.
Okay.
Right, but in the test tube with the hydrochloric acid and pepsin, that's the one that's closest to simulating the actual ideal environment where it's actually doing its job. Right, so it's different. Like amylase is very different. Amylase is optimized to work in your mouth under neutral conditions. So there you put that dude in acid, it's not going to be happy. But pepsin is at its best when it's in an acidic environment.
Okay.
So does that make sense for everybody?
Mm-hmm.
Okie dokie. Let us now go to share screen.
So we ran out of time to do the last one, the cream one.
Did you come up with anything?
No, because we were too busy with the egg whites. We're in a thick discussion about it. So we didn't get a chance. So I'm not sure if that's.
No, I did it. I say it for our group.
You did? Okay.
I just did it. Well, not
Right now.
Not
Okay.
not perfect, exactly.
I mean,
but
what would you think? Would cream and pancreatin break down on their own?
it will be some acid, so it will turn a little pinkish.
Yeah, and why?
Because the presence of pancreatin, that it's an enzyme.
Exactly. So we have the enzyme, we have the fat. probably going to have some breakdown. What about cream with just bile salts?
Oh, I said that it will have, but no, bisalts are emulsifiers.
So they're emulsifiers. Do they actually do actual breakdown? Are we going to actually see fatty acids turning things into?
No.
Yes, a little bit. It's
Oh,
going to
really?
be brownish orange, like a 2+.
Right. So why would bile salts create the fatty acids?
Because they won't create the fatty acids.
Emulsifying keeps the droplets from regrouping, but it's not
Ah.
actually breaking any molecules apart.
Okay.
Things might look a little different because bile salts are, they're brownish. So you might get a brownish tinge because you're adding some brown stuff. But in terms of getting the pink from the breakdown, that's not really something you'd expect unless you're actually doing a enzymatic breakdown of the triglycerides into fatty acids.
So first has to come the pancreatine, break down the fats, and then the emulsifier will keep those droplets apart from each other.
Or
That's
it's more
the
the emulsifier keeps the droplets apart so the pancreatin can get in and has more surface area to act upon.
okay.
Right? I mean, if it's like kind of going back here. This is an important concept. If I have my fat all in a big glob, and let's say this red is the pancreatin, it doesn't have a lot of surface area to act on, so it's not going to be very efficient. If I have lots of little droplets, normally you can't make them stay apart because they just want to come back together. But if you have the bile salts that are then keeping it emulsified, so now maintaining this really large surface area of fat, now the pancreatin can come in and be way more efficient because it has so much more surface area to attack in terms of the fat. So the bile salts make it way easier for the pancreatin to come in and efficiently break down the fat. So does that make it clearer? Okay.
So for that one, if it's really red instead of pink, that means there's more fatty acids.
Correct.
Okay. All right.
Correct. All right. Share screen again. What about litmus cream and pancreatin and bile salts? What would we expect?
Red, like optimal.
This is going to be the optimal, right? We're emulsifying and we got the enzyme. So this is going to be the most efficient breakdown we can have. Like I said, the fact that we have cream means that there's some emulsification. That's what cream is. It's kind of an emulsifier, milk fat stuff. What about cream just sitting with water? Would we expect that to break down much? Not really. All right. So experimental procedure. The first part of it actually was place some saliva in the spot plate and add some acetic acid. This is done just to show that saliva has two parts because the acid will precipitate out the mucins, these little glycoproteins that make your spit kind of gooey. So this first part is kind of showing that your saliva has two things going on. One is the slippery gooey stuff to bind your food into a bolus, but then obviously for the other part of the lab, we'll see there's enzymes to break down starch. So we are gonna look at this go now. Let me. right, and some of these I might go speed up a little bit because of the time, but we're gonna take a look at these.
Okay, we're going to be going over part 1, salivary digestion of carbohydrates. And the enzyme we're using for this part of the digestive lab is salivary amylase. And the source of that is our saliva. So it says to collect 7 mils of your own saliva in a graduated cylinder. So I have close to 8.5, almost 9 mils. And so I've been salivating, collecting it in my little spittoon graduated cylinder. We're going to be using this for not only the spot plate experiment, but also for the 5 tubes of the carbohydrate digestion.
So just, making it really clear, for this experiment, we really are just using spit. This is not something we bought from a chemical supply house. It really is, the results you're gonna see are from Becky's spit, and you'd see the same results if it was your own spit. So this is the quality of your own saliva, besides just being something that looks like spit. It's got some cool chemical stuff going on. So here again is my lab partner saliva that we were using as our source of the amylase enzyme for the carbohydrate part of today's lab. But saliva has a couple of roles. One is helping break down carbohydrates if you're initially chewing up your food, but it also has mucus that helps kind of bind your food into a bolus to make a nice little slippery ball you can swallow down your gullet. And so we can actually see that other aspect of the saliva. If we take some saliva here, taking a mil or so of saliva, and this is acetic acid, it's basically kind of diluted vinegar, will actually precipitate out the mucins, the mucus part of the saliva. All right, so now use a toothpick. We can see, like, ooh, look at that, all those gooey, goopy strings there. Those are the mucins that are the other component of your saliva that are helping kind of bind and lubricate your food. So imagine trying to swallow a bunch of cracker dust. Yeah, it wouldn't really work. So this is what makes a nice slippery ball go down your esophagus. If you look in detail, totally cool gooey goop in there. All right, yeah. So in your lab write-up, you want to describe what you see there, and that there's this mucus that's obvious if you do it. We'll have to mute that for a sec. So that is part of it. Let's go back to this. This next part is going to be looking at the stuff that we've. All right, so now we're going to just. So this is, I'm going to speed through this so you can, I want you to see kind of how it goes, but again, I am aware that we're running on time, so I'm going to kind of speed up and get to the kind of real payoff parts of it. Describe part 1 of the digestion lab. Here, the salivary digestion of carbohydrates. We're going to be using this saliva that was generously provided by my lab mate, and this will be the enzyme. And then we've got our substrate for the enzyme, our starch here, and then we can look at how this process happens under different conditions. So to start, we're going to have to just put some starch. We have 3 mils of starch that we put into each of these 3 tubes. I'm just going to bring this up here. I'm going to go, there's 3 mils in there, 3 mils in there, 3 mils in there. And then also 4 and 5 are going to be controls. Part of an experiment, whenever you're using some kind of an assay, you want to validate your assay. Make sure you give your assay something where you know what the answer is supposed to be, and make sure you get the answer that you're expecting. So then you trust what the answers are when you're looking at your unknowns. So for test tubes 4 and 5, we're going to have test tube 4, which is just going to have pure starch in it. And when we do our test, it better say that, oh, it just has starch in it. Test tube 5 is where we're going to have pure maltose. And I'm going to put that in right now. I'll put that in a little later. And so when we can check our assay for maltose, that better say pure maltose with no starch. All right, so we have starch in our 3 unknown tubes here. Test tube 1 is going to just fill out our volume by adding 3 mils of water. So 1, make sure I can see this in 3 mils of water. We got 3 mils of water in test tube 1. Test tube 2, we have to add 3 mils of this quality high grade saliva. And let's bring this up to 7, so we spit it out. Spit it out into there. For test tube 3, before we add the saliva in, we are going to increase the pH by using this concentrated hydrochloric acid. This stuff is something you got to be careful with. You should not be using this without wearing eye protection. You know, regular glasses can work. If you don't wear glasses, you got to be wearing goggles of some sort. This can damage your eyes, so, and your skin or your clothes or anything. So be careful with it. We're going to add 5 drops of the concentrated hydrochloric acid here. 1, 2, 3, 4, 5. And make sure. So now we have this very low pH solution, and now we're going to add 3 mils of our saliva into this. So 3 mils of saliva, but the conditions here are going to be very acidic for this salivary amylase to try to do. So what's probably happening to that poor amylase right now as you watch?
Being denatured.
Say it again.
It's being denatured.
Yeah, exactly. We've just put the amylase into this super low pH environment. It's not happy.
This job. It's important to remember, keep it in your mind now, that each of these tubes we're going to be testing both for starch using iodine, as well as testing for the presence of the products of digestion, maltose, which we're going to use Benedict's reagent for. So we're going to be putting these in the incubator. And after we're done, though, we need to make sure we take each of these tubes and we split it into 2 tubes. That way we can do the glucose test to look for starch in one set, and we can use the Benedict's test on the other side to test for the presence of maltose. If you forget to do this and all of a sudden put your glucose iodine in here without having split it ahead of time, then you're not going to be able to do the test for maltose. So at this point, we're going to take our tubes here and we're going to have to incubate them at body temperature, and we'll come back in a while and see what happens. So here we see test tubes 3, 2, and 1 are joining the test tubes from parts 2 and parts 3. And we're going to close this puppy up and come back in half an hour or so and see what happens. All right, so these have been incubating at 37 degrees C, and now we can do our assay and see what the results are. All right, so each of these needs to be split into 2 roughly equal test tubes so we can check each sample both for.
Right, this is important. Since we're doing 2 assays on each tube, if you don't split it ahead of time, you're screwed, because all of a sudden you've added the iodine to one, and then it's like, oh, there's nothing left to add the Benedicts to. So you have to, we're splitting each of them. So for each tube, we're going to be able to look for maltose and look for iodine using these 2 different techniques.
Maltose as well as for starch. And again, it doesn't matter exactly, to just be roughly equal. And these are my 3 experimental tubes here. And then remember, I also have the control tubes I have here. This is just pure, before was pure starch. So when we do our test, this should show up positive for starch. It should not show any maltose. This one here is pure maltose. So we do the test for maltose. This should be positive for maltose, but have no starch present. That's how we're going to know our test is trustworthy. So we can start with the test for starch. We use this iodine, and 3 drops, we say. 3 drops of glucose iodine. So I'm going to put 3 drops in each of these tubes. 1, 2, 3. 1, 2, 3. 1, 2, 3. Well, that's not good. Oh, that's good. 1, 2, 3. And 1, 2, 3. All right, so
if you just take a look at it from here, what are you thinking about test tube 1? Is there starch in there or not?
Yes.
Yes. So that black purple means positive for starch. And that's, that hopefully that's kind of what you were thinking, right? It's nothing in there but starch and water. It doesn't break down on its own. Test tube 2, what do we see instead though?
It's amber color, which it's caused by the breakdown of starch caused by the amylase.
Right. So minimally we can say there's really not much starch here because it's not turning blue-black. So that means something happened to the starch. I bet you it broke down. So
if we look for starch, 1 is dark purple black, presence of starch. 2 is amber, that's just the color of the iodine. So this would be no starch, looks like. 3, we also have a very strong reaction here. And this is encouraging. Our pure starch is showing positive and pure maltose is showing negative. To do the maltose test, we have to add in 4 mils of Benedict solution into each of our other tubes. So now we're going to, again, for each tube, let's heat this up. And we'll get, there's 4 in there, 4 in there, and finally, 4, 2, 1. All right, so now each of these is going to have to go and get boiled for 2 minutes. So let's put these in. We have this in boiling water here. We can also make sure, like when you're using a Bunsen burner, make sure you have a nice blue flame that's hot. If it's not adjusted, this is not hot. The water's not going to boil. So you want to make sure you have the blue cone of heat. And so again, just to show you again with the starch, pure starch is going to have this very strong reaction where you get this purple black. And in the case of the maltose where there was no starch present, we can see the iodine just stays this kind of amber color. So this was no starch at all. This is showing presence of starch. And we'll come back in a couple of minutes when the Benedict solution had a chance to do its thing. All right, so these have had their 2 minutes in the boiling water. I'm going to carefully put them back in the rack here so we can see what happened. Here's our controls. 3 and 2, 1. And here we can turn off the fire. And now let's go take a look. So first we can go to our controls again, and we can see this Benedict is blue. Initially, it stays blue if there is no evidence of maltose or any of these reducing sugars. And so this is what we were hoping. This was pure starch in here without any maltose, and this shows from Benedict that there is no maltose present because it's staying just blue. This would be a very strong positive reaction with Benedict where it turns this reddish orange. Again, there's a whole range of colors it can turn. It can turn from kind of greenish all the way to this kind of tomato red orange, and that would all imply the presence of maltose in our case here. So this is great from our control perspective. Starch was negative for maltose, maltose positive for maltose. If we look at our unknowns, this was 1. This was starch just with water. So starch basically on its own after sitting at 37 degrees. Blue means, what does it mean? And why? 2. This is showing positive for Benedict. We definitely got maltose appearing in this tube. We can talk about that in your discussion, why that makes sense. And 3, this one was the same as 2. These were both exactly the same, except in 3 we had that concentrated hydrochloric acid in ahead of schedule before we put in the amylase. So the enzyme ended up having to try to do its job under this really low pH condition, and we can see a very different result. Definitely something you want to think about and discuss.
All righty. So that there you can see. I mean, I know I went through fast, but I think at least for me, I can understand myself when it's going blah 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 blah. And actually, you'll see I actually have the results in a nice little file for you after we're done with the videos. Are there any questions about what you saw there? Right, and you said it's important that the controls turned out correct. Sometimes they don't. Sometimes we do that and all of a sudden we find that there is like some positive for sugar in the starch solution, and it's because somebody's messed up and contaminated the solution by putting the wrong pipette in the jar or something, in which case you can't trust any of your results because you realize your solutions actually are contaminated. So it's really important to do the test with some known and make sure the answers are what you expect. That way you have more of an ability to trust the results of the things that were kind of unknown for you.
I have a question about test tube 3.
Uh-huh.
I don't understand why it appeared cloudier than the other ones.
What do you mean cloudier?
Well, they said to like make an observation about why, you know, how 3 looks different than 1. It's because HCl was added earlier, but it appears cloudier. So
it's well 1 compared to 3 because 1 has just water. 3 has spit in it, which is the mucins, which is going to make it.
the saliva. Yeah. Oh, gotcha. Thank you.
Yeah, so the thing you're looking for is the color. You don't really care about the clarity. The clarity is going to be messed up because of the spit. So, but the color is the thing you really want to pay attention to.
Yes. Sure.
So that, all right, let's see, what time is it? Let's do the lipids.
All right, so here I'm demonstrating part 3 of our digestion lab, and the first part is to investigate the properties of bile salts and how they can emulsify water.
So this first part is also not what you wrote your hypothesis about. This was looking at just emulsification. And she's basically going to take oil and water and mix them together and see what happens. Normally, if you mix oil and water and let them sit, what happens?
They separate.
They separate. So we're going to see how that behavior is affected if you add bile salts. So here we go. This is also something in your lab notebook. You'll describe the results that you see in this video. This is part step 1 in the lipid part of the lab.
And oil. And so here I have 2 test tubes labeled A and B. I have added 3 milliliters of water and 3 milliliters of oil. And so the water is at the bottom and the oil is on top. So both test tubes have the same. What I'm going to do now, I'm going to add some bile salts into test tube B. And so you just need a little pinch. And then I'm going to use some of this paraffin, cut some squares. Just going to put some on top here and put some on test tube B. And then I'm going to shake. So we shake for 30 minutes or 30 seconds, and then we're going to just stop. So I'll show you test tube A and B here. And we can compare them right after I've been shaking. So test tube B, this one has bile salts in it. So we're now going to observe what happens over the next couple of minutes and see if we can get the oil and the water to separate out, which we would expect to happen in A, but let's see what happens in test tube B where we added the bile salts.
Okay, so it's almost 1 minute. So 1 minute, let's take a look at them again.
All right, so we waited a minute. You can see the water and the oil they're starting to separate here in test tube A. In test tube B, we have some foam and some emulsification situation. So we'll wait another minute and take a look at what happens then. All right, so we're gonna take a look at our test tubes now after 2 minutes. You can see at the bottom of test tube B, we have a little water down here. It's colored a little brown, yellowish because there are bile salts also in this water. You can see that the test tube A, we have the water layer and the oil layer coming back. So we'll wait another minute and take a look at what happens after another minute. All right, so after 3 minutes, we can see that we have water and oil separating in test tube A, and in test tube B, we see an emulsification layer here in the middle, and then water down at the bottom, and then we have some foam up here at the top.
So do you all see here? This is oil and water by themselves. After a few minutes, it's basically separated. The oil's back on top, the water is on the bottom. Here, there's this huge range area here, and this is the emulsification layer. This is where you have little droplets suspended in the water phase. And then this has also got kind of just suds on it. This is kind of like a detergent. So it's all getting foamy. But this is kind of giving you a sense of what happens when you mix water and oil with bile salts. They stay mixed together in this big, again, this emulsification layer rather than just separate out into a big block of fat in a block of water. Again, this is why the enzyme is going to be able to be much more efficient because it's got way more surface area to attack. So it's 12:30. Let me, what I would say, this, you can watch this on your own. This is basically lipid step 2 and 3. You'll get to see, what happens as you go through this whole thing. It's going to show how she does it, and incubate, coming out of the incubator and all that. And if we go to the next page, so you should look at these. I'm gonna skip through this right now because of the time. I don't want to keep you trapped here. But Digestive Lab Results. This is showing pictures. So you can use this in your lab notebook, and you can also, in your writing up, kind of see things without having to like worry about the videos as much. Here's the Lugol's test for test tubes 1 through 5. Here's the Benedict's test for test tubes 1 through 5. So you can use this for your data. For the lipids, this shows, again, test tube 1, it is getting pink, which you expected. Test tube 3 is even kind of deeper pink, which is what you expected. And 2 and 4 are both looking kind of blue, which is what you expected. So no surprises here. One of the things you'll see in the protein thing, the protein part actually didn't seem to work very well. You should watch it so you can see what happens, but we don't have results for you here because they weren't really that interesting. Class results for last year in this Excel spreadsheet. This will also be useful in looking at these specific results and putting them in the greater context to make sure that this wasn't just like a one-off mistake. You can say in general, when we looked across a bunch of groups, indeed, like, for, lipids, in general, tube 3 tended to have more digestion than tube 1, and tubes 2 and 4 tended to not have digestion. So it's useful. It's always good to look at your specific results and then look at them in context of, the larger collection of group results to make sure that your results make sense and something didn't get weird on you. So you've got this, we can view, let's make it a little smaller. So this has the data for a bunch of different groups from from last semester, or 2 semesters ago, I should say, as well as the data for the specific results when we just did it in lab a couple of months ago. So, use that data, and say whether it supports your hypotheses, and discuss. So are there any questions about that? Yeah, I think it probably makes sense. It's just as easy for you to sit and watch the video, and because we've kind of discussed the main points. If you have any questions about this, obviously feel free to ask. And I'm going to be in office hours here for the next hour. So if you start launching the stuff on your own and you have questions, you know where to find me. So otherwise, I will see you all on Thursday for another exciting edition. Yeah, three more classes only.
Thank you.
All right. See you all.
Thank you, Professor. Bye-bye.
Bye.
I have a question about the research paper. Since there's so much paraphrasing in it, is it like okay to have, say I'm like describing an experiment, just have like a big chunk of text and then cite at the end, or is it
Better not
proper?
to. Yeah, you really don't want to do direct quotes. It's just not, you know, it's a stylistic thing more than anything, I think, but it's just not what people do.
Yeah, not direct quotes, but if you have like a big chunk of paraphrasing, like multiple sentences, do you want us to
That's not paraphrasing if it's taken directly.
No, not directly. Not directly. I'm just asking about this doing the in-text citation. Do we need to do it at the end of every sentence?
No. If there's a whole bunch of stuff that's coming from the same paper, then you just have to put it at the end of that block.
Okay, I just wanted to make sure because I know like sometimes I have like I'm doing like kind of like mixed sources, but on the same topic. So I have like a chunk and then a citation, and I
Right.
didn't.
So if you're shifting where the things came from and it's not clear, you have to put the reference after like you can't say, I don't know, like, you know, if reference A is talking about, you know, roses are red and reference B is talking about, you know, violets being blue, you have to say roses are red, reference A, but violets are blue, reference B. Whereas if that was in the same paper, like reference C was just talking about the relationship of roses and violets, you could say roses are red, but violets are blue, you know, reference C.
Okay, thank you. I didn't want it to, you know, I know you have like the similarity scores afterwards, and I didn't want it to be like seem weird if I didn't have a reference after something that was paraphrased if it's in the middle of a big block. That was all.
I mean, you just need to, yeah, no, I mean, and paraphrase means you're using your own words, right?
Mhm.
You're not like directly taking chunks. You're kind of taking what are they trying to say and then, you know, writing your own content from it.
Yeah. Okay, thank you.
All right.
I had a question about the presentation.
Uh-huh.
So for the PowerPoint, like we have to reference it. Do we just kind of do it the same way as we did our paper, like with in-text citations and at the very end, just a reference list?
You know, I should even say, typically, you could do it that way. Often in these presentations, they don't even have the reference list at the end. They just assume that if you are interested, you know how to look it up, go into PubMed or something. Right? In a paper, you always need to have them at the end, but in a visual presentation, typically they don't put, because people aren't gonna, they're not gonna write down the full thing. They're gonna write the author and the year, and then it's just easy to find if they
So
want.
then we put the whole citation on the page that we're using or whatever, instead of a reference list.
So you just use like the author or year.
Oh, okay, so no big long citation at the end. Okay,
Correct.
okay, that makes sense. And then I don't know, would like a 5-minute presentation be okay or longer?
You know, 5, if you can say everything you need to say in 5 minutes,
Yeah.
that's great. No more than 10 minutes.
Okay, 5 to 10. All right. And we're going to be starting these presentations on Thursday?
Correct.
Okay. Okay. And then one last question about the lab for digestion. So for the one about protein, egg white, 5 milliliters pepsin, and 5 milliliters water for test tube 1. I'm assuming there would be digestion, even though pepsin likes to work in a low pH environment. Would that be? I guess I have to look at the results.
Yeah, I mean, and like I mentioned, the actual results we got were problematic. Yeah,
Okay. But we still need to write up the results for the protein.
correct.
Okay.
Yeah, I mean, what you're going to write is, this is my hypothesis and this data doesn't seem to support it and what might have gone wrong.
Okay.